Hi, my name is Greg Drolin and I'm a research fellow at the University of Sussex, and today I'm going to talk about some of the work we have been doing to validate our non-coding RNA biomarker signature in the serum of ALS patients. We have been interested into whether we can use small non-coding RNA as biomarkers to help with diagnosing ALS, and while people have previously focused on a specific subtype of non-coding RNA called microRNA, we have been interested in looking at all non-coding RNA. What we have found is not only are there a range of non-coding RNA that can be found in the serum using small RNA sequencing, but that they are differentially expressed in ALS patients compared to healthy controls. So in our discovery cohort of ALS patients and healthy controls from the biomarkers cohort from Oxford, we found 4 microRNA, 1 pRNA and 2 tRNA fragments that were changed in ALS patients. Further, when we used the expression of non-coding RNA to classify the samples in a random forest model, we were able to tell if a sample was from an ALS patient or not 93.7% of the time. To test if the model held up in additional cohorts of samples, we tested it with samples from the ALS biomarker study from London and saw that while the same dysregulation in the expression of our candidate biomarkers was not exactly replicated, there was good performance in the random forest model based on the biomarker samples. However, we did see there was a decline in performance in classifying ALS patients specifically. Further, when we tested our model in samples from Ulm, Germany, we found that the performance in classifying ALS samples specifically was poor at only 10% correctly classified, indicating that our identified biomarkers and the classification model did not perform well. This suggested that non-coding RNA may be susceptible to more subtle variations in sampling, and/or that our biomarker-derived model was overfitting. Therefore, we combined our over 300 samples together and looked at the overall changes in our candidate biomarkers, and we saw significant changes occurring in the expression between our control and ALS groups. When we created a new random forest model using our combined cohort and undertook exhaustive cross-validation with a 33% split 100 times, we observed improved performance, including in the classification of ALS samples specifically. This suggests that there may have been overfitting in our model and that sampling across multiple cohorts and locales improves the performance of non-coding RNA biomarker signatures such as ours. We would like to thank our collaborators and thank the Motor Neuron Disease Association and My Name's Doddy Foundation for their funding and their support.